Hello everyone, and welcome to today's presentation. I'm Dr. S.K. Singh, and I'm excited to share with you insights into liquid chromatography, mass spectrometry, or HPLC, MS. Today, we'll explore how this powerful technique works and its applications in analytical science. So let's get started and dive into the fascinating world of HPLC, MS together. Welcome to slide 2. Here, we're introducing the core concept of HPLC, MS. Essentially, it combines two powerful techniques, the separation step, which is HPLC, and the detection and identification step, which is handled by mass spectrometry. The separation part, HPLC, helps us efficiently separate complex mixtures into individual components. Once separated, the mass spectrometer detects and identifies these components based on their mass-to-charge ratios. This combined approach is widely used across various fields such as pharmaceuticals, where precise analysis is crucial, bioanalysis, which involves studying biological samples, and proteomics, the large-scale study of proteins. A key advantage of HPLC MS is its suitability for analyzing polar and thermally labile compounds, meaning compounds that are often difficult to analyze using traditional methods because they are sensitive to heat and have high polarity. Overall, this technique offers a versatile and sensitive way to analyze complex samples, making it a cornerstone in many scientific and industrial applications. Let's take a closer look at the principle of HPLC separation. At its core, HPLC relies on the interaction between the analyte, which is the compound we're trying to analyze, and the stationary phase, which is fixed inside the column. These interactions determine how quickly or slowly different components move through the system. As a result, components elute at different retention times, allowing us to separate complex mixtures effectively. To facilitate this, a high-pressure mobile phase flow is used. This high pressure pushes the sample through the column, making the separation process much faster and more efficient. Because of this setup, even very complex mixtures can be separated with high resolution. Now, the basic principle behind HPLC involves two main steps. First, the system separates the components based on their interactions with the stationary phase. Then, the mass spectrometer detects the ions generated, identifying them by their MZ ratio, or mass-to-charge ratio. This combination provides us with valuable structural information about the analytes, and the system's high sensitivity ensures we can detect even minute quantities. Overall, HPLC coupled with MS is a powerful tool for detailed and sensitive analysis of complex samples. Let's talk about the interface principle in LC-MS coupling. The interface is a crucial component because it connects the HPLC system to the mass spectrometer. Its main job is to convert the liquid effluent coming from the HPLC into gas phase ions that the mass spectrometer can analyze effectively. There are several common types of interfaces, but two of the most widely used are electrospray ionization, or ESI, and atmospheric pressure chemical ionization, APCI. These interfaces are vital because they directly impact the sensitivity of the analysis and the overall compatibility between the liquid chromatography and mass spectrometry systems. Now, moving on to the core principle of mass spectrometry itself. The process begins with ionization, where the analyte molecules are converted into charged particles. Next, these ions are separated based on their mass-to-charge ratio, or MZ, in the mass analyzer. Finally, the detector records these ions, allowing us to generate a mass spectrum. That mass spectrum is essentially like a fingerprint for the compound we're analyzing. It provides detailed information about the molecular structure and composition. So, in summary, the interface ensures efficient transfer and ionization, while the mass spectrometry process gives us a detailed fingerprint, everything working together to identify and quantify compounds accurately. Let's take a closer look at the mass analyzer principle. Essentially, it separates ions based on their mass-to-charge ratio, or M. This is fundamental because it allows us to identify and quantify different ions in a sample. There are several common types of mass analyzers, including the quadrupole, time of flight or TOF, and ion trap. Each of these has its own strengths, especially when it comes to determining the resolution and accuracy of the measurements. Now, moving to the basic principle, which is closely related. We use ionization techniques that are considered soft, meaning they preserve the molecular structure of the analyte. This is crucial when we're analyzing biomolecules, where maintaining the integrity of the molecule is key. Speaking of ionization, let's focus on the ESI principle, or electrospray ionization. In this method, we start with charged droplets. As the solvent evaporates, these droplets shrink, eventually releasing ions into the gas phase. This process is especially important for biomolecules because it allows us to analyze large, fragile molecules without breaking them apart. So, in sum, the mass analyzer separates ions by MZ. Different types offer varying benefits, and the ionization techniques like ESI help us gently transfer molecules into the gas phase for accurate analysis. This is all about ensuring we get precise and meaningful data from our samples. Now, let's take a look at detection and data acquisition in this process. The detector's role is to measure the ion intensity, which is essentially capturing how many ions of a particular type are present at a given moment. This measurement allows us to create two key outputs: the chromatogram, which plots time against signal, giving us a visual of how compounds elute over time, and the mass spectrum, which plots mass-to-charge ratio against intensity, providing detailed information about the molecular weights and structure of the ions. These outputs are crucial because they enable both qualitative and quantitative analysis. Qualitative analysis helps us identify what compounds are present, while quantitative analysis tells us how much of each compound is there. By accurately measuring ion intensities and translating that data into these visual formats, we can gain a comprehensive understanding of the sample's composition. This step is fundamental in making sense of complex mixtures and advancing our overall analytical goals. Let's delve into the quantification principle. Essentially, it relies on the relationship between peak area and concentration. This means that as the concentration of our analyte increases, the peak area on the chromatogram also increases proportionally, allowing us to quantify accurately. This principle is fundamental in creating calibration curves, which are used to determine the concentration of unknown samples by comparing their peak areas to those of standards. We also incorporate internal standards. These are compounds added in a constant amount to all samples and standards to improve accuracy and compensate for any variability during analysis. The method offers high sensitivity, making it ideal for trace analysis where detecting very low concentrations is crucial. Now, moving into more advanced techniques, tandem MS or LC, MS builds on this principle by using a two-stage mass spectrometry process. In the first stage, MS1, we select a precursor ion, which is a specific ion generated from our analyte. This ion then undergoes fragmentation, breaking into smaller product ions. The second stage, MS2, analyzes these fragments, providing us with detailed structural information. This two-stage process not only enhances sensitivity, but also offers structural confirmation of the analyte, ensuring we're accurately identifying the compound. Plus, it significantly increases the selectivity of our analysis, helping us distinguish our target analyte from complex sample matrices. Overall, these principles and techniques are key to achieving precise, reliable, and sensitive quantification in our analytical workflows. Now, let's talk about method validation according to ICH-Q2 guidelines. When we validate a method, there are several key parameters we focus on to ensure it's reliable and fit for purpose. First, accuracy. This tells us how close our measurements are to the true value. Then, precision, which is about reproducibility and consistency of our results. Specificity is also critical. It ensures our method can accurately measure the analyte without interference from other substances. Linearity is next, confirming that our method provides proportional responses across a range of concentrations. We also assess the limit of detection, or LOD, and limit of quantification, or LOQ. These define the smallest amount we can detect and quantify reliably. Lastly, robustness tests how resilient our method is under small, deliberate changes in conditions. Altogether, these parameters help us confirm that the method is truly fit for its intended purpose, giving us confidence in the results we generate. Let's now look at system suitability and compliance. First, there are some mandatory checks we need to perform. These include ensuring that our retention times are reproducible, which is crucial for consistent results. We also need to verify mass accuracy, meaning our measurements must be precise and reliable. Additionally, assessing the signal-to-noise ratio helps us confirm that our signals are strong enough to distinguish from background noise, ensuring data quality. Moving on to regulatory requirements, compliance with 21 CFR Part 11 is essential. This regulation focuses on data integrity, making sure that our electronic records are trustworthy and tamper-proof. We also need to follow SOP-based documentation, meaning standard operating procedures are our guiding framework for consistent and compliant workflows. Together, these checks and requirements help us maintain high standards and ensure that our analytical processes meet both technical and regulatory expectations. Let's take a look at some of the key advantages of this analytical approach. First, it offers high sensitivity and specificity, meaning it can detect even tiny amounts of a substance while accurately distinguishing it from others. This is crucial for reliable results. Next, it excels in structural identification, so we can determine not just the presence but the detailed structure of compounds, which adds confidence to our analysis. It's also suitable for complex matrices, such as biological samples or environmental samples, where many different substances are present. This versatility makes it a powerful tool across various fields. Finally, it's widely accepted in regulatory submissions, which means it meets stringent standards and is recognized by authorities worldwide. All these advantages collectively support the robustness and credibility of this method, aligning perfectly with our goal of ensuring accurate and reliable analysis in our work. For further support, please feel free to contact us. We're here to help you with any questions or additional information you might need. Thank you so much for your time and attention today. We appreciate your interest and engagement, and we look forward to assisting you further.